Hello and welcome to ResearchPod. Today we're discussing diabetes, a condition that may impact the lives of many of our listeners or those around you, and one that's taught about in science classes as early as secondary school. So it may surprise you as much as it surprised me to learn that we seem to have it all wrong. Professor Michael Schwartz from the University of Washington is speaking with me today about his research into the role of signaling hormones in the brain to affect and maybe even restore the organ functions thought all but lost in diabetes. Michael, hello. Thanks for joining us today.
Thank you very much. Happy to be here.
And by way of introduction, could you tell us all a bit about yourself, what's led you to your research on diabetes, and, well, a bit about your journey through medicine?
Sure. Well, when I was in college at the University of Colorado, which began in the 1970s, I developed an interest in sort of the brain behavior interface. In other words, how does the substrate of the brain result in perceived feelings or motivations, that type of question. And I went to medical school in Chicago after college, and I continued to have that kind of an interest. And after medical school, I relocated to do my medical training at the University of Washington in Seattle. But throughout that training period, I retained that sort of research interest, and I ended up selecting a fellowship in endocrinology and diabetes with a research focus, although it was a clinical fellowship, because there's a lot of interaction between the endocrine systems, which make hormones and so on, and the brain. And so this was an opportunity for me to pursue this sort of brain behavior question with some really talented scientists that could help me develop my research career. You know, when I started my career, obesity was viewed as mainly a problem of willpower, that people who become obese are eating too much, and if they could control themselves better, they wouldn't have that problem. And that was despite a lot of sort of basic science evidence that suggested that body weight is actually regulated through a biological control system. Now, when you say body weight is regulated, what are we really talking about? We're talking about body fat stores. And why is this important from a biological perspective? Well, body fat mass is your body's fuel stores. So in a way, the amount of stored fuel in the form of body fat is related to the amount of circulating fuel in the form of glucose. One is something that you store away for when you need it. The other one is what you're going to use right now. And so it would make sense that the regulation of these two things would go together. And it also therefore makes sense that if you have a problem with one, you might have a problem with the other, which is what occurs in type 2 diabetes.
Now, diabetes is something that a lot of people listening to this may have first or second-hand experience with, and it's covered in a lot of biology classes around the globe. So maybe we can kind of set the scene and recap at first what everyone knows about diabetes before we get into what's new to know about diabetes.
Well, diabetes is a condition that is associated with an elevation of the blood sugar level. So blood sugar or glucose in the bloodstream is the main body fuel, and it's tightly regulated within a very narrow range in normal individuals. And as people develop diabetes, that range becomes impaired and the blood glucose level tends to rise. And over time, having an elevated blood sugar level can cause serious damage to the body. So that sort of defines diabetes, but there are different types. The main distinction between the types of diabetes depends on whether you're able to make the hormone insulin or not. So insulin is made in a specialized part of the pancreas called the pancreatic islets. It's made by a specialized cell called the beta cell. And in patients with type 1 diabetes, your own immune system attacks the pancreatic islet cells and kills the beta cells, so you can't make insulin. You become insulin deficient and the blood sugar level rises. In type 2 diabetes, that's not what happens at all. In fact, exactly what causes the problem is not really clear, but type 2 diabetes is highly associated with obesity. And obesity is associated with resistance to the action of insulin. So your body's tissues that need to respond to insulin become less responsive. And in normal individuals, you compensate for that by making more insulin. But in patients who develop type 2 diabetes, the ability to make more insulin becomes impaired. And so you end up with the combination of insulin resistance and inadequate insulin secretion in the setting of obesity is the classic picture for type 2 diabetes. While all of those statements are true, it's also true that we actually don't know how these things evolve. What is the relationship between obesity, insulin resistance, and beta cell failure leading to inadequate insulin secretion in type 2 diabetes? So that's still an open question, and that was something that became a major focus of my interest over the course of my career.
Now, this would be something for me to edit back into what we were talking about earlier in terms of what led you to diabetes research, that food itself is a particularly emotive topic for a lot of people, and that there's this tangled web between mood and food and emotional regulation via hormones. Well, it's perhaps a cyclical arrangement, or, definitely not just a straight line of A to B to C.
But to get back to your question about the relationship between motivation, diet, and body weight control, what became clear from basic science work and then now has been pretty clearly shown in human obesity research is that it's important to think about the cause of obesity or obesity pathogenesis as having two separate and distinguishable components. So I would emphasize that there is no one cause of obesity. Every individual has probably somewhat different factors. But the two separable components are, on the one hand, you have to enter a state of positive energy balance where the calories that you're consuming exceed the calories that you expend. And so you gain weight as a result of consuming more calories than you're expending over time. So that can happen from any number of reasons. It can be related to how active you are, what type of foods you're eating, your emotional state, social and environmental factors, and so on. But the thing that makes obesity somewhat perplexing is that, as I mentioned, body weight is regulated in normal individuals. And what I mean is it tends to remain relatively constant over time. And if you try to change it by going on a diet and losing weight or overeating for some period of time, your body will correct for that typically, and you'll come back to where you started from. So there are biological processes in each of us that are designed to keep our body weight constant. And in those individuals who become obese, you enter a state of positive energy balance where you're gaining weight. But if you check whether that new weight is being biologically defended, it is. So part of the problem is the defense of elevated body weight. So one problem is gaining the weight. The other problem is biologically defending it. And there's actually been a lot of work done on understanding how that biological control system works. And we're making progress in understanding what goes wrong with it that can lead to obesity. Well, the unfortunate reality is that conservative treatments for obesity tend not to work very well. So this gets back to the brain behavior link. What I started out, my interest, it was, for example, why do you feel hungry sometimes and then you eat and you don't feel hungry anymore? How does that work? And if you try to lose weight or you succeed in losing weight, why do you feel more hungry than you did if you didn't try to lose weight? And we're beginning to understand that the way that works is that there are systems in the brain that control your appetite, and there are signals that are generated by the fat mass that go back to the brain to tell the brain how much body fat you have. One of these is a hormone called leptin, which is secreted by fat cells themselves, but there are other signals that convey this information as well. But the point is that in the normal brain, whether it's an animal or a human, at an unconscious level is being apprised of what the status of your body fuel stores. Are they okay or not? And as the body fuel stores begin to drop, that will be signaled by changes in the levels of, say, leptin dropping, other hormones that the brain is sensing, including insulin, are dropping. The level of blood sugar is dropping if you haven't eaten for a while. All of those signals end up activating neural circuits in the brain that drive feeding behavior. And if you have lost weight from your previously regulated level, that system gets turned on even more. And that activation of that system leads you to feel hungry if you have lost a lot of weight at times when you wouldn't otherwise necessarily have been hungry. And that state will persist until you restore your body fat stores to where they were at baseline. So the problem with current conservative management of obesity is not that you can't lose weight. It's that it's very hard to keep it off, because once you lose it, your brain has a memory of what it thought your body fat mass should be. And it will motivate you to eat more and to lower your energy expenditure until you get back up to that starting point. Now, there are medications that are available that have had a significant impact, but you have to keep taking them in order to keep the weight off. There are also forms of bariatric surgery that can be beneficial to sustaining weight loss in some individuals. But there's a long ways to go for us to say, well, we have a handle on how to manage obesity from a medical standpoint.
Now, to kind of recap on some of what you've just said and expand on neural circuits and their involvement, why look at the brain in the first place when it's something that could and has for a long time been widely considered as just a pancreatic process?
Well, when I did my fellowship training and my subsequent early years of training as an academic, my mentor was a fellow named Dan Port, and he was a leader in the study of insulin secretion in humans and animals. And in fact, he was really the first one to show that insulin secretion is governed in part by endocrine and neural inputs to the pancreas. So it's not simply that the beta cells in the pancreas are responding to the blood sugar. They're responding to the blood sugar in a way that can be dialed up or down depending on other inputs, including from the brain. But I was perfectly happy with the idea that, most of what controls blood sugar is how much insulin is coming out. And how much insulin is coming out is determined mainly by what happens to the blood sugar, and that there's some problem with that in diabetes. And that was the dogma. And so to provide a context for how my thinking about this topic changed, we'll go back 10 or 15 years to work that we were doing in animal models of diabetes. And these are models where there's a drug that you can use to cause diabetes by basically damaging the pancreatic beta cells. So you damage the pancreatic beta cells, within a day or two, the blood sugar goes up very high. And as you would expect, at the same time, the insulin levels drop off to very low values very quickly. That's the whole thing you expect to happen. But the other thing that happens that we realize is that the leptin levels in plasma also drop off very rapidly when you kill the pancreatic beta cells. And that's because the fat cells are relying on an insulin signal in order to make leptin. And if that insulin signal goes away, the leptin goes away. But what we realized is, well, now you have high blood sugar. You think it's due to low insulin, but how do you know that low leptin wasn't also playing a role? So we wanted to answer that question. Now, getting back to the food intake story, there's a long-standing people who have especially type 1 diabetes would know that when your blood sugar is out of control, you tend to be hungrier than normal. And this is called diabetic hyperphagia, or increased food intake in uncontrolled diabetes. And it's very easy to demonstrate in animal models. So we wanted to know what was the role of leptin deficiency in these aspects of what happens when you have diabetes. So we arranged to give leptin back as a subcutaneous infusion over time in rats and mice that had diabetes. And we only gave the leptin back to replace what would normally be there. And what we found was when we did that, their food intake never went up. So the reason that diabetic animals have increased food intake is because of low leptin, not because they had diabetes per se. So that was kind of interesting. But then we wanted to show, we assumed that this effect of leptin was occurring in the brain, because that's where most of the action of leptin occurs. But it hadn't really been demonstrated. So we wanted to repeat the study, but give leptin directly into the brain as an infusion into brain ventricles. And to do that, we infused leptin at a much lower dose than you would need when you gave it into the circulation. And when we did that infusion, we did find that the food intake became normal, as you would predict. But we also saw something else that we didn't really expect, which was after about 4 or 5 days, the blood sugar went from being quite elevated to gradually dropping. And then by about a week, it had become completely normal. So here you took animals that make no insulin, put leptin in the brain, and their blood sugar becomes completely normal after about a week. And that wasn't thought to be possible. How can you possibly reverse the elevation of blood sugar induced by insulin deficiency by targeting the brain? But that's what the data showed. And then when we stopped infusing the leptin into the brain, the blood sugar came back up again to where it was before. So this was the first evidence that the brain can be targeted to normalize the blood sugar of a diabetic animal. And for me, that was kind of a game changer, because this whole idea that it's all a relationship between insulin secretion and glucose handling in peripheral tissues like liver and muscle and adipose tissue, all of that became, wait a minute, what is leptin in the brain doing? The first thing we thought of course, is, well, maybe leptin in the brain is reversing the problem with insulin, and now you're actually making insulin, whereas you weren't before. And we looked very carefully at that, and you're not. You don't make any more insulin when you get leptin in the brain than if you got a control vehicle infusion into the brain. So it wasn't that we were replacing insulin, it was that we were normalizing the blood sugar through a mechanism that was completely insulin independent. In other words, the brain has the inherent ability to normalize the blood sugar of diabetic animals without the need for insulin. That was kind of a game changer for us. Now, of course, we haven't shown that that's true in humans, although we are planning a study to do that over the next year or so, basically to recapitulate what we had shown in the animal models in a collaboration with a colleague of mine named Zaman Mirzada, who is a neurosurgeon down at the Barrow Neurological Institute in Phoenix.
Now, maybe I'm just being overly suspicious, but did you ever face any internal challenge or opposition in terms of conducting your research that, either this would be a dead end of research, or that there's, someone who's making very good money producing insulin over here and don't go rocking the boat?
Well, I wouldn't say that, but I would say that the bar for overcoming skepticism is higher when you're pursuing an idea that is new and is not widely accepted or has not widely been demonstrated by lots of other people, especially if it kind of causes a paradigm shift. The way we thought about blood sugar control and high blood sugar levels in diabetes, in my view, needs to be modified in view of these types of findings, at least based on the animal literature. So we decided to continue pursuing it, but we decided to move over to type 2 diabetes from type 1 diabetes because, number one, type 2 diabetes is much more common. So about 90% of diabetes is type 2 diabetes. And number two, because it's associated with obesity. And we were already doing work on brain systems that are involved in body weight control and that become abnormal in obese individuals. So it might help just to take a moment to go back to what I mentioned earlier about the defense of elevated body weight or body fat mass. In a way, you can make the argument reasonably that as an individual goes from being normal body weight to increased body fat mass to the point of becoming obese, it's not that there's a failure to regulate body fat mass, it's that the defended level of body fat mass is increasing over time. And you can make the same argument about blood sugar levels in patients who go from having normal blood sugar to type 2 diabetes. It's not that the blood sugar control system has failed, it's that in a typical human, this evolution from normal blood sugar to diabetic range blood sugar occurs over 5, 10 years or more. And it's a gradual process where the blood sugar is still being regulated. It's just being regulated at a higher level. So in some ways, the underlying disease process is similar. You have a variable that is subject to homeostatic control, and that homeostatic control continues, but the level that's being defended is gradually rising up. And you could say the same thing about high blood pressure. Normal people maintain their blood pressure in a normal range. People with high blood pressure, it's not that they don't regulate their blood pressure, it's that the level that they're regulating gradually increases to the point where it's abnormal. And from that standpoint, it's kind of interesting that high blood pressure, obesity, and type 2 diabetes all tend to occur together in humans as part of what's known as the metabolic syndrome. So we think there may be some causal link between them and that the brain may be involved. Now, why do I say that? Well, if you look at the brain area, an area called the hypothalamus, that is important in energy homeostasis neurocircuitry, we and others have shown that if you look at animal models like rats or mice, within a very short time, a few days or a week or two, of switching them onto a diet that will cause them to become obese. For example, a diet high in fat and sugar, as compared to their normal chow, which doesn't have really much of any fat or sugar in it. As the animals start eating, they love this diet and they start eating a lot of it. And within a short period of time, you can see injury occurring in this particular hypothalamic area where these neurocircuits are involved. And there are markers of that injury, which are activation of non-neural cell types, so-called glial cells. There are multiple different glial cells, which are basically support cells or inflammatory cells in the brain, and they get activated in this specific brain area. And that occurs not only in animals that are obese, but if you look using imaging methods in humans, obese humans also have this evidence of what we'll call gliosis in this part of the hypothalamus. So these things go together. The human work has been done primarily by Ellen Schur, who's a colleague of mine here at the University of Washington, who does brain imaging in humans. And she found that just as in the animal models, humans also develop gliosis in this part of the brain in the setting of obesity. Not only that, she more recently published a paper showing that this gliosis response is even more strongly linked to type 2 diabetes in humans, and that the degree of gliosis is directly proportional to the impairment of blood sugar control along a spectrum of human subjects with and without diabetes. So this raises the possibility that maybe the same problem that is occurring in the hypothalamus in association with obesity is also somehow contributing to the defense of elevated blood sugar levels in patients with type 2 diabetes. And in some ways, it makes sense because body fat stores are the stored fuel and blood sugar is the circulating fuel. So the idea that they are regulated through an overlapping shared set of brain controls makes a certain amount of sense. And it's a way to at least tie together the observations that we had been aware of. So to move this in a more therapeutic direction, we began studies in animal models of type 2 diabetes, again, rats and mice. And we focused on a family of peptides known as fibroblast growth factor peptides. And there are many of these different peptides, and they are known to have many different functions in the body. But work out of a couple of pharmaceutical companies had shown that if you just administer them at pharmacological doses in these models of type 2, the blood sugar gets better. And it came as a little bit of a surprise. We suspected that that was probably because they're acting in the brain. There's a lot of fibroblast growth factor, or FGF, receptor in the brain, and the types of effects we were seeing looked to me as if they were involving the brain. So we set out to study whether these different fibroblast growth factor peptides had anti-diabetic effects that were mediated in the brain. And we have focused in recent years on peptide known as fibroblast growth factor 1, or FGF1. And we found, kind of to our amazement, that in a particular mouse model of type 2 diabetes, that if you give FGF1 directly into brain ventricles, a single dose one time, the blood sugar will drop initially. It continues to drop over about 24 hours or so till it reaches normal. So it goes from being quite elevated, maybe 3 or 4 times normal, down to normal. So that was exciting that it seemed to be working in the brain. And this was a relatively low dose of FGF1. But what was even more amazing was that the blood sugar just stayed normal well after the FGF1 would have disappeared. So it came as a surprise, but we ended up just monitoring the blood sugar of these mice. And it turns out that even if you go out 4 or 5 months, the blood sugar is still normal after a single injection. So that, again, was a sort of unprecedented finding that the brain can be targeted in ways that with a single dose will reset the blood sugar level from being diabetes range to normal, and it will maintain it there over time. So that's quite different from insulin treatment of diabetes. Insulin will lower the blood sugar, but it doesn't reset it at normal, it just lowers it. You have to titrate the dose to figure out how much to give, and then you have to keep giving it, because if you stop giving it, it goes back up to where it was. What we're talking about here is a single dose of a peptide given into the brain that causes the blood sugar to come down to normal and then just stay there for weeks or even months without having to do anything else. And to make this point even more clearly, it's not simply that you're lowering the blood sugar, it's that you're resetting the defended level of blood sugar, because the blood sugar will stay at this normal level over long periods of time. And if you try to change it, for example, if you give a bolus of glucose into the circulation, the glucose goes up as you expect, but it comes back down to where it was. And if you give a bolus of insulin to lower the blood sugar, it'll go down as you expect, then it'll come back up to where it was. So you haven't just lowered the blood sugar, you've reset the defended level of blood sugar at normal. What this means, again, is that the brain has the inherent capacity in diabetic animals to reset the blood sugar to normal. And that would be of great therapeutic benefit to patients with diabetes, to simply reset the blood sugar at a normal level rather than having to manage every day, check your blood sugar, give a dose of this drug or that drug, do it again the next day, check your blood sugar, which is how diabetes is treated now. So we do feel that this is an important project, ultimately with potential to change how diabetes, not only how it's understood, but how it's treated.
And now I'm kind of waiting for the other shoe to drop. Give me a reason to not get my hopes up too high, to not get too excited about this. Because it can't be this easy to just completely shake the foundations of biology, right?
Well, within the last year, we advanced our studies to inject FGF1 into the brain of diabetic monkeys. And that was done in collaboration with Peter Havel at the University of California, Davis, at their primate center. But unfortunately, the doses that seemed to be effective in lowering the blood sugar also seemed to make the animals feel sick. I mean, they basically stopped eating and drinking for a couple of days. And so we decided we're going to put that on hold until we either figure out how to prevent that effect of FGF1, or we figure out how to do what FGF1 is doing without causing the sickness with some other intervention. So this doesn't have to be all about FGF1. It should be about understanding what it was that FGF1 did, and then seeing if there are other ways of accomplishing the same thing. So let me tell you a little bit more of a story about that work, because I think we're making progress there. What we found is there is a set of neurons that are important not only in food intake control, but in blood sugar control. They're called AGRP neurons. And I won't get into the details of why they're called that. But when they're activated, they drive feeding behavior, and they can also raise the blood sugar level. And if you inactivate them, in diabetic animals, the blood sugar level will come down, at least somewhat. And so we became interested in them as potential targets for the effect of FGF1. That the idea being that FGF1 may inhibit these neurons, and maybe that would happen in a sustained manner that could help to explain the sustained reversal of diabetes. And so we began a series of experiments, and we found that, lo and behold, the brain area that is mediating the effect of FGF1 to lower the blood sugar is the same brain area where the AGRP neurons are located. Number two, we have shown that in an acute setting, administering FGF1 either to a brain slice or to a living mouse will inhibit the activity of the AGRP neurons. Number three, if you put FGF1 into the brain of a mouse model of type 2 diabetes at a dose that normalizes the blood sugar level, and then you wait 2 weeks and then go and look at the activity of the AGRP neurons, they're still inhibited 2 weeks later. So there was no precedent, that you could do something and see it last for 2 weeks. But the fact that that seems to be happening suggests that perhaps the effect of FGF1 to normalize the blood sugar in type 2 diabetes animal models is mediated by long-term inhibition of these AGRP neurons. Now, that's important for a couple of reasons, and I wouldn't say that's the only thing. It's probably multiple neural circuits whose function is changing, but it's important for a couple of reasons. One is that it simply raises the possibility that if you could find some other way to inhibit AGRP neurons in humans, you might have beneficial effects. And I do think that that's a therapeutic direction that's worth pursuing. But it also suggests that the only way you could get an effect that lasts for 2 weeks, I think, is to do some sort of structural reorganization of the circuit. In other words, this can't just be happening to the neurons themselves. It has to be happening to the environment in which the neurons live. And so that gets back to the glial cells that I talked about. And we published a paper in Nature Communications about 2 years ago with Tuna Pears at the University of Copenhagen, showing that when you give FGF1 into the brain of these type 2 diabetes mice, you do have effects on AGRP neurons, but by far and away, glial cell types are much more responsive to FGF1 than the neurons are, especially a cell type called the astrocyte, which is a major glial cell. And it regulates neuron function in a whole variety of different ways. So we think probably the sustained inhibition of the AGRP neurons has to do with effects on astrocytes, but that remains to be proven. We're getting funding to examine that question. So the point is that all cells in the body are bathed in what's called extracellular fluid. And the extracellular fluid is not really a fluid, it's more like a gel or a matrix. And the extracellular matrix contains all kinds of biologically active molecules that are regulating cell function from the outside. And there's a particular type of extracellular matrix in the brain that is called a perineuronal net, where elements of the extracellular matrix actually form a physical condensation like a mesh around neurons. Not all neurons, only some neurons. And this has been studied mostly in other brain areas like the hippocampus or the cerebral cortex. But with Zaman Mirzada, we reported in Nature Metabolism in 2019 that most AGRP neurons are actually enmeshed by perineuronal nets. So that maybe suggested there might be a way to regulate the activity of the neurons by effects on perineuronal nets. And then Kim Alanji, who trained with me as a postdoc and is now a junior faculty member of our program here at the University of Washington, showed that in a rat model of type 2 diabetes, the abundance of perineuronal nets around the AGRP neurons is reduced. And when you give FGF1 to fix their diabetes, because FGF1 works very well in those rats to normalize their blood sugar, you also restore their perineuronal nets to normal around the AGRP neurons. And she even showed that if you use an enzyme digestion method to get rid of the perineuronal nets in the hypothalamus at the time that FGF1 is given, the ability of FGF1 to sustain the normalization of blood sugar is blocked. So, in other words, the ability of FGF1 to have a sustained anti-diabetic effect seems to require its effect to rebuild the extracellular matrix around these neurons. So, and the extracellular matrix is being determined partly by the glial cells. So there's a connection between the FGF1, the glial cells, the extracellular matrix, and sustained changes in neuron activity that result in remission of diabetes.
At this point, it sounds like there's more happening in the brain than there is in the pancreas, or, well, anywhere else.
It's complicated, yeah. But it's worth pursuing because it ultimately helps us to understand not only how normal blood sugar is controlled, but what causes diabetes to occur, what is driving the elevation of blood sugar in patients with diabetes, and can we target that process to provide relief to patients with diabetes in ways that would be less invasive and less demanding than current diabetes treatments.
So, what changes with this new knowledge, and is there anything that is going to be coming out, any more projects about it that we can be looking forward to?
Right. So, I am a physician, and an important goal of my research, in addition to sort of improving understanding of, how body systems work, is that we can bring this information to improve treatment options for patients with obesity or diabetes. And so that work progresses. I think there's a long ways to go because we're just really beginning to peel back the layers of our understanding of what the brain is doing with regard to the control of blood sugar. But there have been some translational, developments, I guess you could say. One of them is that in collaboration with my former trainee, Zaman Mirzada, who is a neurosurgeon who works at the Barrow Neurological Institute in Phoenix, along with Earl Hirsch, who's a colleague of mine and an expert diabetologist at the University of Washington. We are developing a study plan to test a hypothesis that I mentioned earlier that came out of basic science studies. And so the idea is that we know in rats and mice that if you have sort of uncontrolled diabetes due to destruction of insulin secreting beta cells, if you simply infuse the hormone leptin directly into the brain of those rats or mice, the blood sugar after a couple of days will come down to normal, even without any need for insulin. And so, that was important not only in identifying, the brain's ability to control the blood sugar, but it was also the first sort of evidence that uncontrolled type 1 diabetes, or diabetes where the problem is you don't make any insulin, can be treated effectively without using insulin. There was really no precedent for that, certainly not in the clinical literature. So as a proof of concept experiment, we have developed a collaboration and have a proposal that is under review at the FDA in the United States, which you have to have approval from the FDA to do this kind of study, where we're going to be bringing well-controlled patients with type 1 diabetes into an intensive care unit type of clinical environment and actually inserting a catheter directly up into the base of the brain of these individuals on a temporary basis to infuse leptin directly into the brain of humans and then gradually withdraw their insulin, with the goal being to determine whether type 1 diabetes patients can maintain normal blood sugar without insulin by providing leptin directly into the brain. So, the timetable for that really depends on when it gets approved by the FDA, and I don't know when that will be, but I would imagine we'll begin on that sometime in the next year. So that's one example of a clinical translational dimension of our work. I'll give you another one that also involves my colleague Earl Hirsch. And this has to do with the standard of treatment for hospitalized patients whose blood sugar is elevated, especially in the intensive care unit. So this is a very common problem. Patients who are really sick often have high blood sugar levels. And for about 20 years now, the standard of practice for all comers who come into the ICU and have elevated blood sugar, whether they have diabetes or not, is to infuse insulin, usually intravenously, to the patient to get the blood sugar down to a normal level, with the idea being that normal blood sugar is better than high blood sugar if you're sick. And that makes sense, of course. And there is evidence that that is true among patients who did not have diabetes beforehand. But what's becoming clearer now from work, a lot of studies now in hospitalized ICU patients, is that if you came in being diabetic already and your blood sugar was already elevated to begin with, and the first thing you do is to normalize the blood sugar, in other words, bring it all the way down to normal levels. Well, if your body and your brain were used to having a blood sugar that's elevated and you suddenly lower it, the outcome can actually be worse. In fact, it's quite the opposite. So in patients who do not have diabetes, if you look at mortality outcomes, when you normalize the blood sugar, the mortality is reduced. But if you look at patients who had pre-existing diabetes and had elevated blood sugars at home before they came in with an acute illness, the effect of normalizing the blood sugar was to increase mortality. And that's an important finding because it suggests that the extent to which you control the blood sugar in a hospitalized patient in the ICU should be determined in part by whether they had elevated blood sugar to begin with or not. And so you could raise the question then, well, why would a patient who is used to having a blood sugar that is elevated have a worse outcome if you suddenly normalize the blood sugar? And the work that we have done suggests that part of the reason that patients with type 2 diabetes maintain a high blood sugar is because the brain thinks that that's the normal blood sugar. The brain is sensing the blood sugar and it is maintaining or contributing to the maintenance of an elevated blood sugar. At least that's a hypothesis that we're continuing to explore. But the point is that if that were true and you suddenly drop the blood sugar into the normal range, that's going to be perceived by the brain as being too low. And when the brain thinks the blood sugar is too low, it activates responses such as activating the adrenal medulla and causing epinephrine levels to increase. And if you're in acute illness, having an increase of these types of stress hormones may be maladaptive and could contribute to increased mortality. So we're actually in the process of submitting a commentary to the Lancet that addresses this concern and calls for a reevaluation of how do you identify the glycemic target in a hospitalized patient, and should a pre-existing diagnosis of diabetes influence that decision? So we don't know the extent to which that clinical application is a reflection of the role of the brain in control of blood sugar and in the development of diabetes. But my suspicion is that that will be found to be the case. Yeah.
Think about everything that this does to challenge the previously understood function of sugar processing in the body, putting some pretty big challenges at the door of what people have spent years and careers thinking that they understood professionally. Is it time to rewrite the textbooks?
Well, I think the history of science is that the truth eventually wins out. And, I'm not really interested in viewing my work as being somehow in conflict with mainstream thought or having to fight a battle to win acceptance of our ideas. I think, there is still some skepticism in the community about the extent to which the brain is really important, and the onus is on us to make the case, not only in animal models, but in the clinical world as well. So as we begin to make progress in that area, eventually people will have to accept that, you have to accommodate the role of the brain in our understanding of blood sugar control and diabetes. And that's a process that will take time. And, I'm committed to continuing to work on that. I do think it's important for the diabetes community and the rest of the world to be aware of these ideas and their implications. And just to address your specific question about rewriting the textbooks, I actually have been asked to submit a chapter on brain control of blood sugar to the textbook of diabetes. And that is basically, we submitted that, and that's basically in press as we speak.
Is there anything that any patient listening to this could take away to maybe participate in upcoming trials if they are near a center that's recruiting, or if there's anything that practitioners should know? You've mentioned the hospital admission, sugar level stuff that's coming out in a new commentary. Is there anything that they can take into practice immediately, or any upcoming publications that is going to lay things out for them to catch up on?
I think it's safe to say that the neuroscience of metabolism, if you want to call it that, is a highly integrative, multidisciplinary field that is really just emerging. And it's a bit problematic in the sense that most experts in diabetes and metabolism are not experts in neuroscience, and most experts in neuroscience are not experts in diabetes and metabolism. But those fields are going to converge. They have to, because that's the only way we're going to understand these problems. And it's an exciting field, and it's a great opportunity for young scientists because it's both extremely important, but it's also wide open in terms of opportunities. And I would just add on that if there are members of the audience listening to this now who are interested in possible training in our program in Seattle, we'd love to hear from you. So, what about the treatment of these disorders, diabetes in particular? How does all of this inform our thinking? And I've sort of touched on this earlier, but I'll just reiterate that most drugs that are taken today for the treatment of type 2 diabetes are designed to lower the blood sugar for the period of time during which that drug is in the body. And then as that drug wears off, the blood sugar goes back up again. Certainly, insulin is the textbook example of this. You take insulin at a dose that will lower the blood sugar, you may bring it down into the normal range, and then that effect wears off as the insulin is cleared from the body. In other words, these treatments are not reestablishing normal blood sugar control. They're simply dropping blood sugar potentially into the normal range for a while, and then you have to administer the drug again. The new version that we see unfolding that involves the brain is the potential not just to lower the blood sugar, but to reset the blood sugar at a normal level and have it be maintained in normal or near normal range for long periods of time, potentially indefinitely, with interventions that don't necessarily need to be given every few hours or every few days or maybe even every few weeks. In order to get there, we need to identify the neural targets that are responsible for establishing for the brain what the defended level of blood sugar is going to be. And we know using interventions like putting leptin in the brain that we talked about, or putting FGF1 in the brain that we talked about, as we work through basic science to understand what the targets are that those agents are acting on, we can then begin to identify targets that might be attractive for diabetes treatment. In other words, once we know what the neural circuits are, we may be able to target them selectively using medications that will correct the abnormality in their function that is promoting elevated blood sugar levels and allow the blood sugar to come back closer to normal in a more naturalistic way that can be sustained over time. But the cautionary note here is that going from someone who is used to having a high blood sugar to all of a sudden having a normal blood sugar as an abrupt transition, maybe we should be thinking twice about that objective, especially in individuals who have underlying illness like cardiovascular disease or being at risk of a heart attack, which many patients with diabetes are. So that would be one point. especially in the setting of a hospitalized patient or a patient in the intensive care unit. I think some caution is warranted in terms of deciding what you think the target blood sugar level should be. And if the person had been accustomed to having a high blood sugar level before they came in, I think the idea that they're going to benefit from suddenly normalizing the blood sugar may be mistaken. The longer term goal, I guess, would be to be open to the possibility that the brain might ultimately be more effectively targeted in the treatment of diabetes than it currently is. And I think we're already headed in that direction because one of the most widely and effective used medications for type 2 diabetes now are the family of drugs that act at the receptor for a peptide known as GLP-1. These are these so-called GLP-1 agonists, and there's a whole series of these GLP-1 receptor agonists that are being generated and that have long durations of action and or can be mixed with other molecules to have a kind of a super potency. And some of the effectiveness of the GLP-1 agonists are mediated through peripheral actions, like directly on the insulin secreting beta cell to make more insulin. But others of the effects are mediated in the brain. So even though people may not think about the brain as being an important target for these GLP-1 analogs, they are in fact having significant effects on the brain that promote both weight reduction and improvement of blood sugar control. The upshot of the work that we and others have done have led us to an understanding that we already knew that stored fuel in the form of fat and circulating fuel in the form of glucose are subject to tight biological control. But what we're now learning is that the brain is playing an integral role to control both of them. And that's not surprising because, the stored fuel and the circulating fuel are sort of two sides of one coin as far as the body is concerned and as far as the brain's own needs for fuel are concerned. Moreover, we know that the neurocircuits that are involved in controlling energy balance and body fat stores are overlapping with those that are involved in the control of blood sugar, suggesting that they're really part of one large integrated brain control system. This doesn't mean that the pancreas and peripheral tissues are not important. They are. They're all sort of part of one integrated system. We're also learning it appears highly likely that brain's ability to sense relevant signals related to both stored fuel like the hormone leptin and circulating fuel like the blood glucose level is critical to the proper functioning of this integrative system. So why does this matter? Well, because it's easy then to understand that if there was a disorder in the brain system controlling body fuel stores, it wouldn't be that surprising that that same disorder could leak into the neural circuits that are involved in blood sugar control. And you'd end up with a problem of having both excessive body fat mass and elevated blood sugar levels. Even though both are still being regulated, they're being regulated at an abnormal level. And that's actually a pretty accurate description of type 2 diabetes. 90% or so of patients with type 2 diabetes are also obese. So you have a situation where the fat mass is elevated, the blood glucose levels are elevated. And we have growing evidence that the ability of the brain to sense and respond to the signals that control body fat mass and that control the blood sugar are compromised in individuals with type 2 diabetes. Certainly in the animal models that we study, this appears to be true. I think we can be optimistic that the future is bright for patients with diabetes, that as progress is made in understanding how these systems work, new medications can be identified that improve outcomes and make the disease easier to manage for affected individuals.